There it is. Okay, so 5.1 is the beginning of our unit. So the first thing we do is we start to talk about the phenomenon. Every single unit that we have has a phenomenon. This particular one is dealing with race. So it's saying that race has historically been used to separate humans. So we are coming off of unit 4, which is genomics. We just had a big discussion about how we basically are segregated the races and things that we've sort of done to these races, scientific racism and whatnot. It's a very powerful conversation. If you had that in Unit 4, I'm not there yet, but I will eventually get to that thing. It's one of the most powerful days that I teach. But this is sort of going from that and moving a little bit more into, is race a social construct, or is race genetically, are there genetic components? And we'll kind of talk about that as we sort of move through. So it starts off with this sort of view. And so this is like the first day. And so we kind of start here and we talk about, can you tell somebody's race by looking at them? And so you kind of ask the students if they believe that they can look at somebody and tell what race they are. And I really like these pictures because it's difficult, right? We like to put people into little boxes and say, this person is this person is this person is this. And that's not the case, right? And as time goes by, we become more and more, you know, almost the same. Like there's a lot of variance here, but the variance in a human being is very small, right? We are 99.9% identical to every other human being on the planet. And that is the smallest number of all species on our planet. It's because we're young and because we probably got down to a population of about 10,000 when a volcano exploded, about a long time ago. But these ideas that we are changing and we are so different is not really the case. But I will say, as we sort of move through this, we don't want to ever come off as saying that races don't exist or that we don't value races, because there's definitely value there. But what we're talking about is, do we look at a person and say, just because you're this, you're this? And that is what we try to avoid and get the kids to understand. Oops. Okay. So there's a little race literacy quiz. I don't actually do this, but it is here. We would rather give you more than less. So this is the first thing that we do. It is an article that the students read. There are kind of two ways to do this. By this point in the year, for me, I feel like the students have read a lot of articles. So they have read a whole bunch. So I did this thing, and you can choose to do it however you want, but I created an Edpuzzle version of it. And it is literally me reading the article to them with the questions embedded. So there are two ways you can do this. The first way is going to be, just go here, going to this document. And as you go to this document, they read this little piece, and then over here, they're writing down kind of summaries or answering questions. I just did this a different way where I read to them. And for some reason, they love it. They love it that I am reading to them in this sort of weird form. So you can kind of see, hey, it's me reading. That's terrible to hear your own voice. But there you kind of go, right? So I read it and we go through, and then you can see every single one of these, if you're familiar with Edpuzzle, is a question. And so it stops, they answer the question, and then they move on to the next piece of the text. And then it also gives me an opportunity to sort of talk my way through it with the students, and it becomes a thing. They tell me my voice makes them fall asleep, which is, I don't know if that's good or not. Does it mean it's calming? I have no idea. But it's here and they seem to enjoy it. So we start the video all together and then they mute theirs, and then it basically pops up as the questions at the same time, and then they answer them and we move on. And you can assign it right into Canvas or Google Classroom, however you do it. But the article is basically about how we as human beings like to put people in those boxes that I was mentioning. And this is probably the biggest piece of it. These three gentlemen are all three the first individuals to ever be sequenced genomically. So the top is James Watson. That is the same James Watson that is Watson and Crick. Um, he is controversial. He is interesting, but there is James Watson. He sequenced his genome in 2007. He was the first person that was in charge of the Human Genome Project, the government project in the 90s that was sort of going through and doing the Human Genome Project. Craig Venter, he worked for a company called Celera Genomics. His whole thing was competing with the government project to sequence the genome first. He sequenced his own genome. Down there at the bottom is, I'm totally going to butcher it, but Songjin Kim, who was born in South Korea. He was the first Korean individual, and he is a big time person in genomics and genetics. And so what you do is you kind of ask the kids what they think of as far as who is going to be more genetically similar. So they looked at their human genomes and which ones of them had the most genes in common. And what they will tell you is, of course, James Watson and Craig Venter, because they're the two white guys, which I think is really interesting because by white guys, we're only talking about skin color. And that's not what they're talking about at all, right? So they're not talking about skin color. They are actually talking about facial features that come together to create a face that makes us think of a white guy, right? And then down here, we have Kim, who's the only one that is born outside of the country. And so if you look at the little Venn diagram over here, you can see that between Kim and Watson, there are two genes in common. Between Venter and Kim, there are two genes in common. Between Watson and Venter, there is one. And so it kind of leads this discussion on why is that? Do we really know that much about how they all link together? And this is a fantastic quote. It basically talks about that two people of European descent may be more genetically similar to an Asian person than they are to each other, which is exactly what the article talks about. And it starts our conversation about what is genetics and how did we get these skin colors. And then we end, Scott's going to talk about this in a bit, we end with the race, looking at why skin colors are different. This is kind of how we should start framing it. And so race is just a bunch of traits, right? Your hair color, your eye shape, your face shape. Those things may or may not have an impact on anything. They probably don't, right? So what we're finding out is that it doesn't have a lot of impact. One of the things that I heard my whole life, my father is Mexican. And so I always heard that because I was Mexican, or half Mexican, that I was going to end up being a diabetic because everybody in my family was diabetic because we were of Mexican origin, which is not the case. What you find out is if I do get diabetes, it's because of the fact that my father had diabetes and I have aunts and uncles that have diabetes and so on and so forth. It's more of a family thing than a race thing. And so we need to start talking about ancestry, who was born to who, and so on and so forth, and how did those genes get moved down the pipeline. And this is a perfect time to talk about the cultural responsive stuff. And it shows up in almost all of our units, definitely in all of our units, but almost all of our activities. And this one it does right away. It's something that scares a lot of people. So just kind of keep in mind that when you start talking about race and stuff in the classroom, it seems scary, but once you kind of get in and do it, it's a pretty amazing experience. As I said before, the sort of Socrative thing we do in Unit 4 is one of the most powerful things we do, and it just kind of leads into this. But the kids definitely have to have that good relationship with you before you want to try to do a lot of this stuff. All right, 5.2, Scott Davis.
All right. So any questions about that first task set before we move on? All right. So this one, moving into 5.2, focuses on how actual populations change over time. And it's really when they first start exploring really what is natural selection, what are some of the factors that are associated with natural selection. And if we have an example, how can we determine if it's natural selection or not? And if you've taught, I don't know, I think in Hillsborough, a lot of our middle school life science classes will sometimes do the HHMI Rock Pocket Mouse Simulation Lab. It's been around for a long time. Some people would call this an oldie but a goodie. But basically, when we're looking at it at the junior level, we are doing, you kind of have options as far as what to focus on. Option A, if you look at the Rock Pocket Mouse Interactive with the slides, and if you want to click on that. So this is, if you look at the top, this one is where they actually look at the frequency of the alleles and how that changes over time. So I have actually done the allele frequency version of this before. It's definitely more in depth, and it provides a good connection to the genetics part of it. I know somebody mentioned earlier, Jason teaches AP Bio, I teach AP Bio. Somebody mentioned, about taking some of these activities and modifying them for AP Bio. AP Bio has a lab that's been around a long time where students do different simulations with allele cards. And this would be a great way to plug that in as an AP Bio lab. And I know when I first started doing this during CDL, I did the option B version, which is where the students look at color illustrations of different environments in the Sonoran Desert down in the southwest part of the United States, and they actually count the number of mice that have light colored fur versus the number of mice that have dark colored fur in different locations. And there's a video component. As far as I know, every HHMI activity I know of has some sort of a video introduction. This one's relatively short and sweet, but it's in my personal opinion, it's a very good introduction to what's going on and gives them the kind of the scenario of you have light colored sandy environment that is changed in one area based on volcanic eruption producing darker lava rock. So it's getting students thinking about how does the environment affect the change that we see in populations and how is that related to natural selection. This is what Jason's got up here right now is a Desmos version, Desmos Classroom, if you've ever used that. Used to be called Teacher Desmos back in the CDL days. But this is a Desmos version where students can basically go through the same thing and do the counts, and they put in how many light colored mice, how many dark colored mice, and it goes ahead and graphs it for them so they can actually see what's happening to the number of light colored mice versus dark colored mice, and it produces the graphs. There's a couple of different, if you go to the slide that has the two graphs on it, let's see. There are a couple of different graphs if you use the Google Sheet spreadsheet to plug in your data. There's the bar graph, which you see here, and there's also an option on the patterns version for a line graph. There it is. I'm getting ahead of myself. So, and the nice thing about the line graph is you can go back to what we talked about months and months ago related to carrying capacity. So the line graph actually helps students see, oh, not only is there a change in the phenotype based on a change in the gene caused by a change in the environment, but there's also a limit regardless of how many mice can actually exist in this particular area. So the other thing that's nice about this particular lab, if you go back one more, the other thing that's nice about this is that it gives them a chance to start thinking about whether or not this is actually an example of natural selection, because there are other examples that will come along later in this unit where they will have to determine, is this an example of natural selection or is it not an example of natural selection? So I will tell you for option A, if you're having the kids do option A, which is the part where they actually determine allele frequency, the students actually, it gets them a chance to get up and walk around the classroom, but we have these two cards. One is big A, one is little a, and they're associated with the amount of melanin in the mice fur associated with the MC1R gene, the melanocortin 1 receptor, and they go around and do mating simulations with other students and keep track of what their genotype is. So you can see an actual change when you type in the data into the spreadsheet. You can see a change in the allele frequency. So that would be a way to kind of take this a little bit deeper. And in my classroom, when we came back from CDL, we tend to focus on one activity, one task set in each of the units as our big summative project for that unit. And when we did the allele frequency version, that was the big summative project, adding that mathematical depth to it. We don't, it's probably important to note at this point, we focus on allele frequency, but we're not asking them to do like Hardy-Weinberg calculations. So if you look in the NGSS standards, that is not part of Hardy-Weinberg calculating, is not part of the standards for that. We do that in AP Bio. So if I was doing this lab in AP Bio, I would definitely have them calculate a Q and a p-value and do all of that. But if you're just doing the allele frequency with your students, it's just a matter of figuring out how the dominant allele and frequency of the recessive allele changes over time based on the environment. So
Is this the lab, Scott Davis, that you use to do with like a little swimming pool?
yeah, we actually ordered a miniature blow-up swimming pool, and we took a bunch of cards with these letters on them, put them in the pool. So after each mating and the students recorded what their genotype was, but then they would become the offspring that they produced. And so if you became little a, then you would go to the pool that had all these cards in it, and you would literally pick out two little A's, and then you would walk around the room with your cards behind your back, and you and your partner would each donate one allele. So it's a nice kind of retro tie-in to Unit 4 when they're learning about all of the Mendelian rules of genetics. And so it was a nice way for them to actually see that happening. And like I said, it gets them moving. And they actually kind of thought it was pretty funny that we had a gene pool in the classroom. So if you're looking, if we go back to, let's see, version A, or excuse me, version B. So version B is the version where they just look at the different environments and count mice. So that does not include allele frequency. I've actually gone back to doing the version B because we're focusing more on the human skin color as our big summative at the end of this unit. And so, and to be honest with you, if you're pressed for time and you need to do this in like one day, option B is probably the way to go, because you can print out those color photos and have the kids at their desk count the number. The Desmos version that Jason made, very quick and easy to get through. There is, if you'll note at the bottom of the version for option B, there's a share and trade activity. And what students do is they basically write a summary of what they saw based on the data that they collected and graphed. So they, to the best of their ability, in one sitting, they write down what they saw in a summary, and then they meet up with another student in the class and basically trade sentences. The goal being to try to create this perfect paragraph. It's not necessarily a CER, but it's similar to that in the sense that they're coming up with a claim and using evidence from their graphs. And so that's what they mean by share and trade summary. You're writing down what you think is your best guess, best summary, and sharing it with a partner and getting information from them. So yeah. So if you go to the slide, the Learn Genetics slide, Jason, with
Here?
no, on the webinar slides, just let's go forward here. One more. There we go. So one of the things that you introduce during this activity are the 3 questions that we use this website a lot. HHMI has a lot of great stuff. The Learn Genetics has a lot of great activities, especially case studies. So what we're doing here is showing students a way to objectively determine if something is actually natural selection or if it's not natural selection, because there will be some not natural selection examples later in the unit. So if they can answer yes to these questions, is there a reproductive advantage? Is there variability? The variability in this case being the gene that produces different amounts of melanin in the mice that create different fur colors. Is that trait being influenced by genes that get actually passed down from parents to offspring? So if you can answer yes to these, and the focus being on the individuals that have a trait that is reproductively advantageous, being able to successfully reproduce more offspring, then yes, that is an example of natural selection. All right. Any questions about that? Pretty.
Yeah, Scott, you got a question about, is the gene swimming pool activity in the unit outline?
It is based on option A, and I don't think we added the actual like gene pool to the procedure.
Do you
But
have a copy of yours that you could put in the chat
yeah,
during
let
this
me
next
see if I can
section?
find it.
Okay, 5.3. So we are going to start talking about adaptations. The one thing that you're going to kind of notice, and it's how evolution works, is that evolution takes a long time. So trying to find examples of evolution not taking forever is difficult, which is why Rock Pocket Mouse is used so often, is because it takes place on a pretty small time scale in relationship to other evolution. The other thing that is very quick at evolving is bacteria. And so this is a thing that we're looking at, is antibiotic resistance of bacteria. And so in the course of a lifetime, the first antibiotics came out in the 1920s. I think it was 1928. And since then, these antibiotics have lost their ability to function because the bacteria is evolving so quick that it's making those antibiotics no longer functional. And so that's kind of the whole point of 5.3. We start looking at that. So it starts off with this NOVA video called Hunt for Nightmare Bacteria. I usually show it. It's fairly short, and it kind of just shows an experiment where they show how fast that bacteria is able to evolve so that it can get closer and closer to the antibiotic. And then you use this stronger and clearer protocol. I kind of wrote down what that is here. A student writes a response, a claim or whatever they are creating, and then they share their response with a partner. Their partner then gives them feedback on it, and then together they bounce it back and forth as they create a stronger and stronger, hence stronger and clearer protocol. This is also a place where we highlight pharmacy techs and pharmacists. Actually, a couple of weeks ago, I was talking to a pharmacist and I was telling him about a project that I'm working on that I'm going to mention here in a second. And he was so intrigued by what we were doing. He was just like, that's amazing. And so he was able to give me some information and some things that I was using. And so there are some career highlights. This is a good point to talk about, that in the summer, PMSP does put on what are called externships. If you've never done one of the externships, they are pretty great. And what they do is they take you to businesses around. Unfortunately, it is in the Portland metro area at this point, but you basically go. I've been everywhere. I've been to Intel, I've been to Clearwater Services, I've been to a tool manufacturing place, and it's all places that are requiring science and math backgrounds. And, we keep trying to bring in more and more and more. So kind of look for those to pop up and take them. You do get paid for them, which is cool. You get paid to go hang out at a business, and I think they're pretty neat. I've seen Scott at a few of them.
Oh yeah, they. We started doing this before COVID, and I still remember The best one that I've ever gone to was at OHSU, where they actually work on showing how they sterilize surgical equipment. And they're just begging for students. It's an entry-level position that potentially will lead them to other positions in the hospital. And there's money for continuing education and certification. So definitely a worthwhile thing.
You can see again, we are looking at, is this actually natural selection? So we're doing that exact same rubric that Scott just went through. Did they say yes to all three questions? And if they did, then yes, it is natural selection. If they said yes to two of them, but not one of them, then no, it is not natural selection. This is what's in the works. So this is my big project of the year, and I'm doing a couple of these. My goal for all of these units is to have an inquiry lab and an engineering lab for every single unit. This particular unit and unit 4 are super difficult because of the constraints of trying to create DNA or genetic things or make things evolve. So my thought is, and the thing that I've been working on is to use a Flynn lab that looks at antibiotic resistance. So you take those little, they almost look like paper cutouts with antibiotics on them, and they create the circles where the bacteria only get so close to them. And so what I want to show is that over the course of time, those circles have been getting smaller, right? So since 1995, whenever a certain one came out, I want to show that it's getting smaller. So my thought is, and what I'm trying to do is to look at the research, and if it says that it's 95% less effective, I want to turn that into what would happen if you did this lab in 1995? You know, how big would that circle be in comparison to the fact that you just did it today? And so that is kind of what we're working on. So just like the race and skin color lab that we're going to talk about in a minute, this is in the works. I'm going to have my kids do it this year. And then after this year, I'll put it out there for everybody to use. I'm also creating one for Unit 4 that is some electrophoresis stuff. So you should see some of that stuff start to pop in.
All right. Any questions about that one, Tessa? So
If you have, yeah, but by the way, I put in a plug. If you have something you're like, oh, I do this and it's awesome and it really fits with what you were just talking about, send me an email. My email's at the end. Be like, I have this. This is what I've been working on. Like, I would love to collaborate with you.
Mhm. All right, we are on 5.4. This is where we take a turn to looking at the process of speciation or macroevolution. How do new species evolve? And this kind of like the Rock Pocket Mouse lab, this is an oldie but goodie that you may recognize. There's a couple of different speciation labs that I feel like are floating around out there. We have this one. I've also done a version of this involving, I don't know if any of you are familiar with the California salamander lab, where they look at an actual like map of California, and it's a paper pencil type activity. So it was not very conducive to CDL like this one would be, but it's a similar experience for them, looking at how does the environment affect speciation, and kind of emphasizes the idea of reproductive isolation as being the tool or the mechanism for speciation. So the phenomena here is there are about 150 species of Anole lizards that live on the islands in the Caribbean, around the Caribbean Sea. Before they jump into the actual HHMI lab, the students will interact with the Tree of Life, which if you're going to Google this, the link will take you to the right spot. But just a thing to be aware of, if you Google Tree of Life, you're going to get a bunch of stuff that's not necessarily scientific. So you want to definitely include OneZoom. It's from OneZoom, as you can tell in the upper left-hand corner. The nice thing about this is that it does include domains like Archaea. So if you're teaching an IB or AP course where you need to include some information about classification, you could definitely incorporate this into your curriculum. A lot of teachers will have students type themselves in, like Homo sapien or human, I don't know, Jason, do you want to type that into there? You can type in whatever organism you want. In the upper right-hand corner, type human or Homo sapien, and it will take you to common ancestors. It's got the bonobo, the chimpanzee, so it's pretty cool. Let's see. It reminds me of, if you've ever used Prezis, it's very Prezi-like. So there we are. So, and the students can use their mouse to move around and see common ancestors. We've got 3 different species and where branching would have taken place. It's a good time to, if you're going to incorporate anything about cladograms or phylogenetic trees. So after they look at this, there is a video from the Learn Genetics, going back to that website again, just a nice overview, quick overview of what really is a species and what are the biological requirements for something to be a species. Like, how do we even know if speciation happened between 2 individuals? So they watch that. And then on the next slide, they jump into the reproductive barriers activity. And this is one area where you could incorporate the INB and have them take some notes. You'll notice in the actual patterns curriculum, there's not a ton of note taking. So this is one opportunity to include some note taking in the INB on different reproductive barriers. And then there are 4 different modules in this particular HHMI lab. So if you're just wanting to focus on this particular task set, we suggest doing module 4, which is focusing on the colors of the dewlaps, the little skin folds that come out underneath their mouth. And the idea is it's basically, as the students work through this virtual lab that they can do on their Chromebooks, they figure out pretty quickly that the dewlap is like a flag to woo females. And so they go through module 4 and they end up actually collecting data by measuring using a color scale from 1 to 6, the brightness of that dewlap. And obviously, it's primarily used to woo females. So if you have a dewlap that is going to be more reproductively advantageous, you're going to meet more females, have a higher reproduction rate, and a higher survivability, pass on more of your genes. And so there's if Jason, if you want to click on the student template, they end up going through, answering the questions, and also typing in data that they can use to automatically create graphs. There's a Google spreadsheet that students will use. And what the Google spreadsheet does is it automatically gives them the opportunity to calculate without actually having to calculate it themselves. Things like standard deviation, SEM, if you want to include the statistics that you need to teach in an AP or IB course, this is another opportunity to include that. Otherwise, in a general bio class in 11th grade for the curriculum here, the website, the spreadsheet's automatically calculating that. So they're just basically looking at 2 specific, not all 150 species, but 2 specific species of these lizards and looking at the brightness of their dewlaps. And on a scale of 1 to 6, they can go ahead and type in the numbers and collect the data that way on the Google spreadsheet. And it's pretty, if you look at the data set there, there's a pretty dramatic difference between the 2 species. And so once they put that data in and they graph it, they can see that one has a dramatically brighter dewlap than the other. And as they go through the lab, they realize that the environment connection here is that the one that has the brighter dewlap tends to live in more shaded areas on the island and is better able to be seen by potential mates because of the brighter dewlap. Whereas the one with the darker dewlap doesn't necessarily need that reproductive advantage because it lives in an area where there's less shade and more sunlight. So it not having a brighter dewlap is not necessary for it to woo females in the same way as the other species of the lizard that lives in a different environment. So you could actually look back and tie this into the rock pocket mice, talk about, what is the difference in what's happening here, kind of make a distinction between microevolution, change to the gene pool versus change to the species. The other thing to keep in mind if you have time and you want to, is module 1 actually has them measure the length of the lizard's toes. And they eventually figure out that the toe length in the lizard is related to where they live. Do they live in the trees, on branches, or do they live on the ground? And so it's kind of another opportunity for students to look at the idea of how does your environment affect the traits that you end up having and passing down, and how does it determine whether or not you're reproductively successful or not. Because we're trying to get students to understand really what causes speciation is this idea of reproductive isolation, or keep organisms that are not able to reproduce with each other successfully for whatever reason. It could be habitat differences, timing differences, the structure of the organism themselves. So yeah, any questions about this one? It's pretty straightforward, and one that, like I said, you may have done before. If anybody wants to, I do have the California salamander one where it looks at differences in rainfall and climate and how that affects different speciation of California salamanders. So, all right.
Okay, 5.5 is looking at the evidence for evolution. So there's a couple of activities you can choose from here. So there's this first one, it's called Teach Genetics. Everybody starts with a video. So we start with a video on the evidence of evolution, and then we go down here, we do the Teach Genetics fish. And so we're looking at common ancestors. So kind of like Scott was talking about with the tree, we're looking for why do these links exist. And so this one involves using seed cards. I've never actually done this particular version of it. I usually do the one that's on the next page that I'm going to show you in just a second. But this one, I have done something with animals in general in AP where we take a bunch of animals and kind of put them on our own little tree, and we create those with little cards. And so if you print these out, you can kind of see they are the really nice looking seeds. And you would print these out and you could put them out on your table. And I've used string or even the chalk markers to create those links between, and then say what those links would involve. Like, why are they related to each other? There's also this evidence for evolution stations. This one, you are splitting the groups into one of these. And so they are probably going to look at, I mean, that's the way I would do it. You could have them do the whole thing if you would like, but I would have them split up. So they would go through and they would write down, what does this have to do with common ancestry? The second group would answer this. And I kind of like to have those discussions where they kind of get back together and talk to each other about. And then there's that cladogram there at the bottom with transposons, which are genetic genes that move from place to place. This is the one that I think is the coolest. It's the tale of two pandas. The idea of this one, and again, I would split this one into groups. It is very detailed and very well done. It's a lot for one kid to do or one group to do. And so what they do is, again, video at the beginning, and then you jigsaw this case study data, and it's kind of split into pieces already. So everybody kind of reads this little piece up here at the top. But the idea is that you have these two animals. You have the red panda and then you have the giant panda. And the difference between them, the red panda is thought to be a descendant of the raccoon or be closer related to the raccoon, whereas the panda bear has always been thought to be a bear. I mean, it has bear in its name. So trying to figure out like where those links come from. And so you split them into groups to determine whether or not these two are in the same family. They are both called pandas, so you would assume that they are both in the same family, but they determine it. And what I think is really cool about this particular lab is, I'll kind of show you what they do. So here are the splits. And so this is the first group would do these things here. They look at the front paw anatomy, they would look at the skulls, they would look at the diet. And then group two would look more about the holistic, like the gait of those particular animals and their anatomy. They looks at their brain. Then down here is the DNA. This is kind of how I did it in college, where you start splitting them by the DNA components or the chromosome components, which are group four. They're going to be looking at those. And then the hemoglobin sequences, fossil record. Probably the hardest one of all of them is the fossil record, but they would do all of that. So they come back together and they fill out one side or the other for this particular. So do they think that the skull anatomy is more sharing ancestors with the bear or sharing ancestors with the red panda? Front paw anatomy, do they think that's more bear, red panda? So they end up having their little note sessions on these sides, and they kind of go through, and it kind of shows that science is not fully figured this out yet, right? The kid gets to understand like that it is still a work in progress, and they determine what they think. Do they think it's more red panda or more bear? And they might do that by counting up the ones on the side, but I think it ends up being very similar. And so even then they can kind of decide what they think. And so they create a claim and evidence piece from looking at this. And so I would have them present to each other. And so when they present to each other, they kind of go back and forth and kind of create this definition. And so you can see here at the bottom, there is a place where they're going to write just a claim and an evidence, no reasoning in this particular case. So why do they think that it is more like a bear or like a red panda? And that is that one.
Any questions about that? I did make a note. Since you're talking about pandas, Saturday is National Panda Day again.
I did not know that.
Yeah.
How do you celebrate National Panda Day?
Yeah, I don't know. It's combine. You combine it somehow with St. Patrick's Day.
Okay, because tomorrow is Pi Day, right?
Exactly.
Yes, Valerie.
Oh, you're muted.
Can you unmute?
So I've taught the stations one the past 2 years that I've done this.
Mhm.
And I don't know if you guys eliminated it or changed it, but I feel like there used to be part of that claim and evidence and reasoning at the end where they had to actually construct their own cladogram of how they split off from the do you know what I'm talking about?
This particular activity, the one that I'm showing.
They had to there was like a part where they had to make their own cladogram and showing where they branch off from each other, where the red panda
Oh,
and
okay.
the-
There
where they all
is.
okay. So that even as their teacher, I'm like, because you said even the cladogram part was the hardest one, and I don't really even know how to lead them through that because I'm like, it's hard to put them together with their common ancestors in that it seems like there's not enough information in that section to really go off of to feel really confident in the cladogram that's produced. So I was hoping maybe you could kind of talk me through that for a second.
How about I do this? So what I'd like to do is, can I just add to the actual thing and then, because I think you have a good point, right? So the whole idea of the cladogram is very difficult, and students don't have any idea of how to do it really.
Mhm.
But if I could maybe even create a key or something that I could put in there so you could see it and then kind of, but as I said, there's no real right answer on this one, right? So you're kind of, it could be one or the other and you're basing it on evidence.
Yeah.
So the links between them, obviously the ones that are closer related are going to be attached to each other.
Yeah. And then they have to incorporate somehow they had to do the panda, the red panda, the raccoon, I believe, and then the two common ancestors that they were
Yeah.
talked about. And that's the part. Yeah. So if you could show, yeah, that'd be helpful.
Yeah. So what I'll do is I will fix the instructions.
Okay.
So I'll just
That'd
put
be
that
helpful.
on there as something that I'm going to do this week.
Okay. Thank you.
Yeah.
And since we are talking about cladograms, I did have, I think it might have been during the pandemic, a student teacher um, trying to introduce the whole concept of cladograms, because this is an area likely students have never even probably heard of that word before. And so she thought it would be a good idea to take something a little bit more familiar, concrete. So she created a little hostess cladogram project where students had to make a cladogram first based on different hostess treats. Like, do you have, is it chocolate? Is it sponge cake? Does it have filling inside? All of that. So it's just a quick little fun thing I just put in the chat. So, but I agree that in general, cladograms is something that I think students struggle with, even if it is pretty straightforward. Just the whole idea of how to set it up and where to put all of these people and things or objects or whatever it is you're trying to diagram. So this is the culminating activity. Most of what you see on the Patterns bio website includes activities or task sets that have been around for maybe a decade or more. And this is an activity that really, I think we started really working on in the maybe last year, intentionally looking at the idea of skin color and the phenomenon being that humans can't be sorted into biological categories based on their skin color. And yet, when we look at the entire planet, we see all these variations among skin color and realize that there is a health connection to that as you dig deeper. This is also a great way to tie in the discussion at the very beginning. It's almost like the biological race discussion, talking about race being something that we have created as a social construct as opposed to being a biological basis. This is a great bookend for that discussion, and kind of takes it a step further. So if you go to the essential question, we're looking at how did variation in human skin color there. It's based on a case study, but there's also a video. If you've noticed, every time we have an HHMI activity, there's a beginning video, introductory video at the start of whatever the activity is. And this is the same way. So what we start doing at the beginning is we watch a video which is based on some pretty groundbreaking work by Dr. Nina Jablonski, which you may or may not have heard of, but she started doing some study related to looking at some reproductive or selective advantages of different skin colors around the world and what causes that variation, and how can that affect your health, and in what ways does that affect your health. And so the video is a great introduction. I will warn you, when you're watching the video, unlike the rock pocket mouse and the salamander, this is heavy duty. There's lots of detail. They're talking about, they're kind of going back to hopefully what they had in patterns physics in 9th grade about reflection versus absorption, and looking at the role of melanin, and talking again about that MCR1 gene that we touched on with the rock pocket mice. And so you have some options for that first video, because like I said, it can be overwhelming. And I know some teachers will just play the video and let the students answer the video questions while you're watching the video. Jason's got up here an Edpuzzle that he made that has stopping points with the questions embedded into the Edpuzzle. And I know there's also a video, a version of the video that actually has built-in stopping points. It's not on Edpuzzle, but it's just a version of the video where it's almost like bookmarked. So you have options. The video
I also
question,
shortened it quite a bit, so it's
a
a
lot.
pretty
Yeah.
long video, so I shortened it. There's a lot of times where Jablonski is walking around campuses, just kind of hanging out, and I'm like, I'm gonna cut that out just to save some time because
Right. So if you go through, the kids answer questions related to the video. If you look at this, I really like this on page, I think this is 2 that he's showing right now. It really gives the kind of goes over the idea of an umbrella, the way melanin protects our DNA in the nucleus. And kids start to understand that melanin isn't just something that maybe having more melanin makes your skin darker, but it also acts as this little protector, so to speak, of our DNA. And they do talk about the idea of having melanin protect your DNA and keeping your incidence or rate of skin cancer lower, but that's really not the focus of what this project is all about. So it's interesting when you're talking about melanin, that your body uses that as a defense mechanism. You can talk about the idea like whenever you tan, your body produces more melanin to add more protection to your DNA. And so the idea as the kids go through here is they look at a couple of different things. They look at where humans are located on the planet and how that affects the intensity of the UV light that hits them. And question 29, if you go back to the document there. So there's what you can consider either attention, or sometimes I know me and my colleague here at Liberty, we talk about the trade-off. There's a tension between the need to protect ourselves from UV radiation, but there's also a benefit, and the students understand that, learn to figure out that, oh, UV radiation is something that actually is needed in order to produce vitamin D, which we need for healthy teeth and bones and to prevent getting the brittle bone disease, rickets. There's also the issue of folate that the students look at, or folic acid, which is tied to fetal development. So lack of folate is going to result in a higher potential for like one of the graphs that we have focuses on individuals with less folate have higher rates of problems with the development of their spinal cord. So kind of the key pieces here, and the one thing that I would suggest if you're going to do this in your classroom is to look at, kind of create some sort of little quick chart on your board. If you look at the slide that we're looking at now, I actually went next door and took a picture of Miss Marvin's diagram that she created, just as a quick reference, along with this diagram that Jason's had that he made. It's a drag and drop. But the idea is that if you have darker skin, you have more melanin, but that skin also tends to block UV light. And if you look at the diagram on the left here, UV light can penetrate the skin, enter blood vessels, and actually destroy folate. And so you end up creating a situation where you have a folate deficiency. And in individuals that have lighter skin with less melanin, they are able to make more vitamin D because more of that UV light, the UVA light, is actually able to penetrate the skin and be used to create, basically take cholesterol and turn it into what will eventually become vitamin D. So there's this constant trade-off between you need skin that is dark enough to maintain enough folate so that your offspring can develop correctly and increase the numbers of your species, but you also need to be able to have light enough skin with less melanin to allow yourself to make more vitamin D. And so it creates a conversation about, why do we see all these different skin colors? What happens? What is something that I would need to do if I had a certain skin color in a certain area? Would I need to take vitamin D supplements, or would I need to have a diet that might increase my folate consumption or folate amounts? And so if you look back at the CER at the end, if we can go back to that. Oh, one more thing. One of the things that the other teacher here at Liberty and I do, when we look at the data and all of these different graphs that you see in the data analysis for part 3, we actually took the data and I printed it out and gave it to groups of students, like 3 or 4 students, and we had them make data posters. And so they actually took the data that's in this document here and focused on just one graph. It could be like how much vitamin D do different people have, even just at different points in the United States, like somebody that lives in Florida versus Portland, Oregon. What is the difference that we see in amounts of vitamin D that are in those individuals, and individuals with different amounts of melanin that live in the same area. So it gives them a chance. They create a data poster. They put their data up somewhere in your room. You could even turn it into a board meeting if you want to do it more that way. And I'll put there's the template there. So the data posters, these are the graphs that we have in the actual task set. So I print these out in color and I just cut the paper in half and I give each group a graph of my choice. And then they tape the graph to the upper left-hand corner of their poster paper, or you could have them, like I said, use a whiteboard and do a board meeting. And they just talk about, hey, what am I seeing in this data? And then the idea is that they're going to take this data and come up with a reproductive advantage for having a particular amount of melanin in your skin or different skin phenotypes. And what is the take-home message based just on that particular piece of data that they're getting from the graph? And then you have options. I know some teachers like to actually have students take turns with actually presenting their data poster to the class. And then while they are presenting, other students can be filling out their the boxes in part 3 that are asking for summaries of each of those data sets. The other option, and the thing that I do in my class, because different groups finish at different times, so it's kind of hard for me to have the time and the logistical situation for students to do presentations. We basically just do a gallery walk. And so students, we will put the posters up, and when you're done with your data poster, then you'll go around and look at the posters of others and get the take home message from each of those. So otherwise, this is one of those things that could end up taking a lot more time than you might have expected. I tend to do the video with the video questions and the introduction of the whole project on the very first day. And then we focus on the data. And like Jason was showing, we have interactive components where they actually look at different skin cells with different amounts of melanin, and they drag and drop vitamin D or folate into those cells, which is kind of a nice way for them to really start thinking about, okay, if I have a cell with high pigmentation, what does that mean in terms of the amount of melanin? And the melanin is intentionally shaped so that they can put it over the little orange dot that represents folate, because melanin, like I said before, is representing kind of a little parasol or umbrella that's protecting that folate. So if you
You're kind of
have,
seeing behind the scenes here. There's quite a few comments you can see.
Oh,
We've
yeah.
been working on this quite a bit.
Yeah,
I think this is also a good time to say that any of the stuff that we do in any of our units, again, we're going to give you more than you need. Please cut them down to your own, needs. Because if you were to do this full on, you're talking 3 days, maybe 4. And
Right.
so I know my version takes about a day and a half. I try to cut it down as much as possible because usually I'm kind of fighting to try to get to unit 6 by the end of the year. And that's just kind of how that works.
And this is for me, for my unit, this is the culminating summative project. So we do, like I said, video one day, data posters on day 2, and then focus on the CER on day 3. And if I have time, kind of a cleanup day for day 4. One of the things I think you can think about is how you want to word your CER. There are different options. If you look at the version that's posted there on patterns, it's asking them to write a CER based on the prompt, how do the different shades of human skin color demonstrate evolution by natural selection? And here at Liberty, we kind of parsed that out a little bit to make it a little bit more directed. So our version of the CER, which is based on the same data that you're seeing here in the patterns version, so we're all using the same data, is getting students to basically use the data to explain what's the selective advantage, because we're really talking about reproductive advantage, kind of going back to that whole concept of speciation and variation and natural selection. You can use the natural selection questions for this same activity. But our CER is, what's the selective advantage of having low pigmentation, like lighter skin, in the southern and northern latitudes? So if you live north of the equator or south of the equator, what is the advantage of having less melanin, lighter skin? And then what is the selective advantage of having more melanin or darker skin near the equator? And so students really have to, in order to do well on that CER, they have to identify the folate component and they have to identify the vitamin D component. And as you watch the video, like I said earlier, there's kind of a tendency for students to immediately jump to melanoma and skin cancer when you're talking about UV light, and the whole idea about, well, people that live at the equator that might have darker skin, they have more melanin, so their DNA is protected by the melanin that's the umbrella, and that's why it's a reproductive advantage to have more melanin, because you get less chance of melanoma. And she mentions in the video the idea that if you're talking about reproductive advantage in skin cancer, most skin cancers like melanoma develop after reproductive age. And so it's really not the factor that's really at play here if you're talking about it in terms of what is giving that particular group of people or individuals with that particular phenotype, what is giving them a reproductive advantage. If they have less skin cancer or more skin cancer, all of that happens post reproductive age for most individuals. And so if you've passed away or become quote unquote less fit from something like skin cancer, it really should not affect whether or not they have offspring or what their fitness level would be in terms of reproductive advantage. So it is kind of an interesting, the piece that she focuses on in the video is that the folate connection, the idea that they have a situation where they are able to have more folate, which is resulting in fewer birth defects, which is directly tied to reproductive advantage as opposed to skin cancer. All right. Any questions about that? Not sure if we covered everything.
Yeah, I think this is one of my favorite things to do. It's very interesting. And I had an instructional aide, an adult, a few years back, a black man that basically in doing this unit and looking at this particular activity was like, wait, I'm supposed to be taking vitamin D. And then he started taking vitamin D and he came back and he's like, I feel way better. And I'm like, he's like, I learned something. I'm like, good, you learned something. So, if he's learning something, I'm assuming that other students are learning things as well.
It's a really, most of us are going to be doing this, toward the end of the school year when the kids are starting to kind of want to check out, and it's high interest, high engaging, very personal. I do want to mention, if you see the UV index map that is shown on this slide, it comes from the European Space Agency, and the one that was on there before was from 2022, spring equinox. So you can actually have students click on that link and it will take you to the website and you can plug in whatever day you want. So you can look at, where is the UV light more intense in what part of the world? A lot of them assume it's at the equator, but this is a good way to visualize that and look at the effective angles as far as the sun goes. But it's kind of nice. It's kind of a cool website just to be able to type in if I want to know exactly what the UV index was on February 23rd of this year, I can type that date in and it'll pop right up. So
This leads to an interesting conversation too, because most of the people that live in Australia are lighter color. And
mhm.
so they start looking at that. They're like, wait a minute, Australia is in the red. Explain to me why it's got a high population of people with lighter skin. And so
Right.
you
And
get to
then
go through all that cool
you
stuff.
can talk about migration, movement of individuals. There's so many different ways to tie this in. And like I said, it is a perfect bookend for your initial race conversation at the beginning.
Okay, so just kind of taking you through to the end, we're almost done. You can see here where the tasks line up. So kind of where they match the standards and the performance expectations. Make sure that if you need access, that you go to this thing to get that access. As Scott said at the beginning, make sure you have your things unhidden so you can actually see them. Spanish folders are being populated. We have a couple people in Hillsboro. One works at Century High School where I work, and she is constantly translating things and putting them into those folders. But as far as Scott and I were talking about this before, when it's all going to be done, like I'm not positive, but we're working on it. The main issue is that we're too efficient at what we do and we keep fixing things. And so when we fix things, they don't get translated immediately. So it's unfortunate, but that's kind of how it works. But since I have somebody here on staff with me that is doing a lot of the Spanish translation, it's super helpful that she's doing what I'm doing, and I kind of show her what needs to be translated. But it's a lot of work. To get paid, the last thing that you have to do is the exit ticket form. And so make sure you click on that exit ticket form and fill out the thing. Say whatever you want about us. I mean, you can say mean things about Scott. I don't know if I could take it, but Scott is tough skinned. As far as 23andMe answers, I've never done it. My sister did it, and she told me like a lot of things. I'm assuming mine is very similar. Yeah. Let me click on the exit ticket.
I talked to my students. My uncle did. We talk about DNA testing, the pros and the cons. Just it always gets their interest peaked because people are seeing commercials for $70 DNA tests and all of that. And so my uncle did a big, huge family history project about the Davis family and offered to basically, pay for everybody to get tested and do the DNA swab. And since he was paying for it, I did it. And I told the students my DNA went on the website, and you're able to kind of, in general, find out where some of your ancestors might have been from based on your DNA. But the thing that I didn't necessarily think about, and I'm sure I signed something at some point, but that your DNA, which is basically your genetic fingerprint, my DNA ended up, I think, on 23andMe, and somebody put their DNA into 23andMe, and it turned out that we were half siblings that we didn't even know existed. That I guess my dad had an affair or something with somebody at some point and had a child, and so we have the same dad and different mothers. And then my uncle found out that his mom had an affair, and the person he thought was his dad, Mr. Davis, wasn't really his dad, and he isn't actually in the Davis family at all. So created a little family drama. So yeah.
Kay Reed asked a good question about the website. The website takes a bit because our web designer right now is sort of putting all of those finishing touches on it. The best way still is to go to the outline and look at the outline and versions of the files that are there. That is the best way to get what we are doing at the moment. So we pretty much create things, and then once we've tested them, we put them out as soon as we finish them. So
And we're actively, you'll notice, hopefully notice that we're trying to clean up the outlines so that we got feedback that sometimes they just feel overwhelming as far as the amount of text and words. So we're trying to clean up and streamline the outlines and put some of those comments that are really just for teachers in teacher note documents. So I know we're kind of focusing first on these big projects like the skin color project. I know we have a whole set of teacher notes for the biodiversity project in Unit 1. So we're trying to pull out some of that text that is not necessarily for students and more for teachers as kind of a notes or background information that you need, supplies, things like that. So hopefully that'll make it a little bit easier.
Yeah, any other questions before? I know you guys are going to be really sad that we might end a little bit early today, but I see tears. All right, if there's no more questions, feel free to log off as you see fit. Thank you all for coming. I appreciate it.
Yes, thank you so much.